thank you very much for the kind introduction, and also I'd like to thank the organisers very much for inviting me. It's great to be able to talk to this audience. So, people are living longer than they used to. It's a trend that started really in the middle of the 19th century in Europe. And it's illustrated here by the work of two demographers, showing this really rather dramatic linear increase in life expectancy in the time since then. So what's plotted here is life expectancy at birth against the year in which the birth took place for the country that was the world leader at the time. So to start with, this was here, it was the Scandinavian countries. Latterly, as the trend's gone on, it's the Far East. But what you can see is this steady increase of 2.5 years per decade. And the various horizontal lines here are the predictions of individuals or organizations about where the trend would top out, what the intrinsic limit on human lifespan would turn out to be. And the point of this paper at the time it was written, and it's still true, is that there's no clear message in the demographic data of where that limit's going to be, and in many places the trend is continuing. Although unfortunately things like the epidemic of obesity in the young may mean that it turns back to some extent in some places or some parts of society. So it's a triumph of human civilization to be celebrated, that living conditions have been so much improved, public health, medical care mean that people are staying healthier as they age, and so they're living longer. And I think Thomas this morning actually mentioned the record holder, lifelong smoker. She actually gave up when she was 119. Jean-Louise Calment, a French woman, she was born in the south of France in 1875 and lived to about 122.5 years old. And she actually met Van Gogh in her father's shop. He sold paint, and Van Gogh used to go and buy it there. And not at all clear what her secret was. I mean, human lifespan's about 25% heritable, and she came from a long-lived family, so that may have been part of it. But it certainly wasn't her healthy lifestyle. So, something to be celebrated, but coming with a major downside, which is that it's becoming clear now that advancing age is the major risk factor for the chronic and killer diseases. So these are the three main ones: dementia and other forms of neurodegeneration, cardiovascular and metabolic disease, and cancer. These are various EU and UK data, and you can see for the two sexes the very steady and dramatic increase with age in the incidence of those diseases. So I think there's an imperative to try to keep people healthier for longer as they age, to compress that period of disability and disease at the end of life. And since aging is the major risk factor, one obvious tactic would be to try to intervene in the aging process itself to prevent these diseases from developing in the first place, rather than dealing with them piecemeal as they arrive. But I think there has been some pessimism and some skepticism whether such an approach would be possible. And part of the reason for that is simply that aging is very complicated. So if you look in any animal, and in this case in humans, at the different tissues as the animal goes through its life, what you see is a complex picture of different kinds of damage and pathology developing in each tissue, presumably in accordance with the kind of insults of daily living that that particular tissue encounters. And it seems rather unlikely that any simple genetic or environmental intervention would be capable of slowing this lot down simultaneously. You would be looking, at best, at fixing each problem as it arises. But that rather pessimistic way of looking at things has turned out to be, at least to some extent, not true. So there are common features of aging that are shared across many organisms. Some of them are shown here, and you've heard about several of them in the talks this morning. So there's a group to do with the genome, either the structure or the way that it's packaged in the nucleus. Various cellular processes, you've heard about proteostasis. Mitochondria can also become dysfunctional during aging, and so can signaling pathways in cells, particularly nutrient-sensing signaling, which I'll say more about in a moment. And then there are various tissue things like cellular senescence, stem cell exhaustion, and also the systemic environment. Tissues can act at a distance to affect each other's aging. So it is a complex process, but there are certain commonalities to what goes on during aging. And therefore, it seems a prospect for interfering at this kind of level with these kinds of processes to somewhat ameliorate the effects of aging. So there are various ways that one could set about studying that. One that's proved productive and very interesting are these human rapid aging syndromes, the progerias. So these are genetic disorders that lead to shortened lifespan, but in both the cases that I've shown here, also very dramatic manifestations of what looks like fast aging. So the one on the left is Hutchinson-Gilford progeria. This is actually a mutation in one of the proteins, the lamins, that wrap around the nucleus and give it its structure and probably affect the structure of chromatin. And this child, I don't know exactly how old the one in this photograph is, but mostly they die by the age of 14 of various forms of cardiovascular disease. And you can see the loss of subcutaneous fat, the baldness, and so on. There are a number of features of aging that are present there. And similarly, Verna syndrome on the right. This is a DNA replication error. It's a DNA helicase that has a mutation in it. It's a much slower developing condition, but you can see that in the 48-year-old lady over on the right, she looks much older than that and has many of the features of aging. So these have been interesting and instructive, but they tend not to recapitulate all of the features of aging. They're segmental. They just have some of the features and not all of them. And of course, what we'd ideally like to do is to slow aging down, because that's what we want to do with people. We want to actually reduce its impact and the formation of these age-related diseases. And that's been tackled in a number of different ways, but one of them is mutagenesis experiments. So people have simply asked, if I mutate the genome of whatever animal, can I produce strains that will live longer? And it first became really clear that this was possible in the worm, Caenorhabditis elegans, which you've heard about this morning. And this is one of the classic examples in the field now, that gene DAF2 actually encodes the single worm insulin-like growth factor receptor. So this is a signaling pathway that senses nutrients, controls metabolism and growth in response to current nutrition. And you can see that this is a hypermorphic mutation, so the receptor's still there, it's just subfunctional. And you can see the enormous increase in lifespan. That's a survival curve for these worms when you mutate the receptor. These are actually data from my colleague David Gems. This was originally discovered in the lab of Cynthia Kenyon. And interestingly, this effect of this whole nutrient sensing network turns out to be evolutionarily conserved, because subsequent work in both the fly and the mouse showed very similar things. So in the fly here, you can see the effect on growth. The mutant fly on the right is a dwarf. This is the mutation in the insulin receptor substrate, Chico, which takes the signal from the receptor on the cell surface into the cell. And you can see that the flies are long-lived. And in fact, the heterozygous flies, the ones that have only lost one copy of Chico, are still long-lived, but they're completely normal in body size. So these are just two different things that the pathway does at different stages in the life of the fly. On the right, we have a Chico mouse. It's lost both copies of one of its insulin receptor substrates. And again, you can see the nice increase in lifespan. That's actually a survival curve for females in both cases. And one of the interesting things about these anti-aging interventions is that they often have rather sex-specific effects. And this particular network tends to extend lifespan more in females than it does in males, for reasons that we don't completely understand at the moment. But of course, we're not particularly interested in lifespan per se. That's just a way of isolating mutants. What we care about is whether these animals are staying healthy as they age. And in both the invertebrates, they clearly are. They retain their mobility, their ability to learn, their tissue structure is better, they have less aggregated proteins. Most of the things that go wrong during aging go wrong more slowly in the mutants. And in this mouse on the right, there's been quite a thorough phenotyping. And as they get older, what we see is that they maintain functions better for longer. So actually, the mutants start life slightly insulin resistant. They look as though they might get diabetes. But as they get older, they actually cross over with the wild types, and their glucose handling is better in middle and old age. They have a better immune profile, more naive T cells, and they maintain their motor function better. They're more agile and stronger. And they showed delayed onset of various aging-related conditions, including osteoporosis. You can see the cataract here on the eye of the control mouse, and also this ulcerative dermatitis on the head and the nape here. About 40% of the controls get that at some point during aging. We've never ever seen a case in these mutant mice. So here we've got a lesion in a single gene, but it's producing this very broad improvement in function in a number of different tissues, implying that we really have hit something about the underlying aging process. I think you'll hear a lot more about this tomorrow, but there's also some indication that this nutrient sensing network is important in human aging. And this is being looked at mainly through genetic association studies, where people isolate families or individuals who make it to advanced ages, 90 or 100 or even more than 100, and ask whether there are genetic variants in the candidate genes in the signaling network that are associated with this increased survival. And there are quite strong indications now that this insulin IGF network is important. This thing here is interesting, FOXO. It's something called a transcription factor. It controls the expression of genes, and it's the key effector of the insulin IGF signaling pathway. And in both the fly and the worm, if you knock out FOXO, the insulin mutants no longer extend lifespan. They need this transcription factor to do it. And it turns out that genetic variants in one of the FOXOs in humans are associated with survival to advanced age. We can also look at people who've lived to a great age and simply ask what is their signaling status for the signaling network. And again, for both TOR, which is the most ancient bit of the network, it's intracellular, it senses amino acids, and it goes right back to unicellular organisms. It's present in yeast. And also the insulin and IGF do show distinct signs of being tamped down in individuals who make it to advanced ages. So we've got this evolutionarily conserved signaling network that seems to be very important. And almost certainly it's part of the machinery that implements the response to an environmental intervention that's been known about for a lot longer, which is dietary restriction. So if you simply put an animal on a diet, you force it to eat less than it would eat voluntarily if it were left to choose for itself, you extend lifespan. And that turns out to be true in an extraordinarily wide variety of organisms. I've simply illustrated some of them here. It was first described in rodents, rats actually, in 1935. It's been also very extensively studied in mice. And recently there's been some very telling work with rhesus monkeys, which has come out in a couple of very long-term publications, because these animals live for 35 or 40 years. So it's a really major undertaking to do a dietary restriction study with them. These are just some of the animals in one of the studies. And what both of these found was that calorie restriction, as they called it, produces a major and very broad spectrum improvement in health during aging in these monkeys. In one of the studies, they also lived longer, in the other they didn't. And there were a number of differences in the way the studies were actually done that could explain that. But what was very clear in both was that almost every aspect of function was improved during aging, and the monkeys stayed largely disease-free. So what I thought I would do is simply tell you about a couple of recent studies that we've done in this general area in my lab, mainly with the fruit fly, Drosophila. First of all, I'd like to talk a little bit about these sex differences that I mentioned. Very often an anti-aging intervention affects one sex more than the other. And then I'd like to talk a bit about drug targets, because this is going to be the way forward. If we're actually going to translate this work into helping people as they age, then we have to address the issue of drugs and pharmacological intervention. So one dramatic sex difference in response to an intervention happens in these flies in response to dietary restriction. So in flies, the way that we do dietary restriction is simply by diluting their food. So the flies are fed on a gel that's basically a mixture of sugar and yeast to give them a protein source, and they suck on the surface of the food and take up the food that way. And if we dilute the food, they don't respond by increasing their feeding rate. So they basically take what we give them. And if you do that and dilute the food going to the left here, you can see this female response in red. You get a very, very big increase in lifespan up to a certain point, and then it drops off a cliff because the flies are starved. Males show a similar sort of response, but much less. The amplitude is tiny compared with that in females. And we've wondered for a long time why that is. Why do females respond more? And two postdocs in the lab in London have had a look at this. So Jenny Reagan and Mobina Chiricha are the people who've really driven this research forward. And what they did to start with was simply to look at the two sexes during aging and say, well, are there any obvious differences in the way that their tissues age that might mediate this effect of dietary restriction? And they found that there was a huge difference in the gut. And this is illustrated here. So what we have are females at the top, males at the bottom, and they're getting older going over to the right. And in both cases, we can either look down on the surface of the columnar epithelium, which forms the lining of the gut, or make a section through it. And you can see that as females get older, this nice honeycomb arrangement, looking down on the surface, goes completely to pot. You start seeing holes in the gut starting to develop here. We've got wound-healing rosettes. We've got clumps of cells dividing little gut tumors. And eventually, by the time the fly's old, it's just a complete mess. And the same if you look at this nice, neat arrangement of the nuclei in blue here. Again, as they go through, it becomes completely disordered. In the males, essentially nothing happens. If you look at an old male, he looks very like a first-week female. So there's this huge difference. And a lot of that's probably to do with the fact that females eat a lot more than males do. They're basically machines for producing eggs, so they have to eat a lot to make eggs. So the gut's doing a lot more in the way of dealing with nutrients. And this may explain this fundamental difference. The one part of the fly that contains dividing stem cells is the gut. And again, if we look at the two sexes, it's very obvious that there's a difference in the stem cells in the gut. We're looking down on a surface view of the gut, outside of the gut here now. The blue nuclei, again, are shown in DAPI, and these red ones are dividing. And females basically start life with more stem cells than males do, and they divide a lot more actively. And females also lose barrier function as they age. So the gut has to absorb nutrients, but it also has to keep out things that shouldn't be getting through it. And females get leakier guts as they age. So the way we do that is with this smurfing test. We feed the flies a blue dye, and if the gut's fine, then it just goes straight through, it stays in the gut. But if it's leaky, it leaks into the fly, and we get the smurfing phenotype. And we looked at two different strains here, an outbred one, which is the one we usually work with, and an inbred, and in both cases, there's much more smurfing in females than in males. They're losing barrier function. And interestingly, dietary restriction does rescue this female pathology. So this is shown here. We've got an objective scoring scheme. We've got dietary restricted flies here, control flies here, females on the left, males on the right, getting older as they go down the slide. And the critical thing is the comparison here. The males basically don't do anything, and these are young females. Here's the pathology in the control females. It's still there in the dietary restricted females, but it's less. And you can see the quantification of this on the right. It's a quite big effect. Now, one of the reasons that we chose to study this problem in flies is that they have a sex determination system that's completely different from the way it works in mammals. There are no sex hormones. So each cell in the fly decides what sex it's going to be depending on its own chromosomal constitution, basically the ratio of the X chromosome to the autosomes, 2 in females, only 1 in males. And that then starts a cascade of transcription factor activity, which does the differentiation of the cell into either the male or the female phenotype. And what that means is that we can produce a fly that's a mixture of two different sexes. We can manipulate the sex determination cascade in a particular tissue, in this case the gut, and give the male a female gut and see what happens. And that's what has been done here. So we have a very regionalized gut in the fly, just as in mammals. This is the front end here, the back end here. And what the two of them did was to feminize the male gut from here backwards. But they left this region, the proventriculus, still male. So this acts as a sort of control that the gut itself is okay. It hasn't been sex reversed, but it's in a male where the rest of the gut has been sex reversed. And we can use it as a token of whether the gut itself has been generally badly affected or whether we're looking at a specific effect of the reversal. And we can then see what happens here. And what we find is, here's the female looking at the surface and the section. Here's the male. These are middle-aged flies looking at the two, and you can see the normal sex difference. She's showing a more chaotic arrangement here, and the nuclei are very disordered. These are the feminized males, and you can see that here we start to see the disorder that we see in the females normally. Up here we've got the proventriculus, and this is completely unaffected in the males. This is this region that hasn't been sex reversed. So this is the control region, and it's fine. So if you feminize a particular bit of the male gut, it develops the female-like aging-related pathology. And they also have the other features. So stem cell activity goes up dramatically or stem cell division goes up dramatically in the feminized males. They start to lose their barrier function, and they show a great deal more pathology over here on the right. So it's definitely going with the sex of the gut in a cell-autonomous way. And that's interesting because the male still isn't eating very much. So it argues that there's something about the female gut that's pre-adapted to the fact that it's going to get a lot of food rather than an immediate response to the food. But of course, what we can do now is say, okay, we've got a male with a feminized gut. What does this do to the responsive lifespan to dietary restriction? And this is shown here. So we've got females in red on the right. This is just a survival curve at a single level of dietary restriction now. So these are the controls. The dietary restricted females live longer. The green are the ordinary males. They show actually no response at all to dietary restriction in this particular experiment. And here in blue are the males with the feminized guts. And they live less long, presumably because they've now got gut pathology that they didn't have before. We think that males live less long than females normally because they have a more compromised immune system. It seems to be an immunity issue that's killing them. But now they haven't merely got compromised immunity, they've also got a feminized gut, nice double whammy, so they live short. But what they show now is a response to dietary restriction that's indistinguishable from that of females. So it looks as though, at least implies, dietary restriction is increasing lifespan a lot because it's rescuing pathology of the aging gut. And I think, like a lot of experiments, this one probably raises more questions than it answers. Can the gut account for sex differences in the response to other anti-aging interventions, insulin signaling, TOR signaling? Is there a sex difference in gut aging in mice and in humans? The gut's been surprisingly ignored as an important tissue that might mediate aging. And is it important for the effects of anti-aging interventions generally, even ones that aren't sexually dimorphic? So I think there's a lot of work to do here, particularly in mice, starting to look more closely at the gut. So I'd like now to move to the second topic, which is this question of drugs. So there are a number of reasons in aging research for being very interested in drugs. One is experimental. So if you've got a drug, you can do a nice timed intervention. You can do a quantitative intervention by varying the dose. And you can very easily also do combinatorial interventions, where you suppress the activity of different targets in a signaling network, which may be very important. It may be that there's more than one underlying axis of aging, and we actually need to suppress more than one thing to really get an optimal output from a signaling network. So we've been looking at this in the fly. And here we have what's really a very simplified cartoon of this signaling pathway that I mentioned at the beginning, the insulin IGF TOR signaling network. So this, as I say, is the oldest bit of the system, the TOR bit that senses amino acids. And there's a very nice specific inhibitor of TOR. All of the drugs I'm going to talk about, by the way, are licensed drugs for human use. These aren't chemical probes, they're actual drugs. And rapamycin's main use in humans is to prevent rejection of kidneys after tissue transplant. It's an immunosuppressant in that particular context. And TOR is right in the middle of this network. It's absolutely key to it. And it turns out that in all three main model organisms, actually all four, yeast, worm, fly, and mouse, rapamycin extends lifespan. So these are the data for females. This was actually done by a postdoc in my lab some years ago, Verna. She now has her own lab at the UCL Cancer Institute. And you can see the dose-dependent increase in lifespan with rapamycin here. It's not very dramatic in Drosophila, but it's there and it's very repeatable. And it can produce big extensions of lifespan in mice. In both species, mainly an effect in females, again. So that's one nice drug that we have to work with in this network, but we were very keen to develop a drug that would allow us to interfere with insulin IGF signaling. And I've mentioned before Chico. This is this insulin receptor substrate that takes the message into the cell, and if you knock it out in the fly, you get a long-lived fly. And because this forkhead box O transcription factor is required for that, it's always been assumed, I think, that this canonical branch of insulin signaling through these two other proteins is what's really important about producing the extension of lifespan. This is true in both the worm and the fly. But the really annoying thing about this pathway is that there are no good drugs against it. There isn't one single drug that's been shown to extend lifespan in any organism that operates against either of these targets or any of the associated proteins in this bit of the network, which is frustrating. However, Chico also signals through this pathway here, this RAS pathway. And RAS is very important in human cancers. 30 to 40% of all human cancers contain a mutation in RAS. So this pathway has really been hammered pharmacologically. There are many, many drugs against its various constituents. So we became interested simply in whether this pathway was important in producing the increase in lifespan in the fly when we knock out Chico. And we got a hint that it might be from the work of this person, Nazif Alik, who, again, ex-postdoc who now has his own lab, who was doing some work on FOXO. Because if FOXO is required for increased lifespan, then presumably the pot of gold is buried downstream of FOXO somewhere. So what genes is it binding to, and what genes are changing their expression in response to this transcription factor? And that's what this study was about. And one of the things that he found was that there was massive feedback in the pathway. So FOXO talks a lot to genes in the insulin signaling pathway itself. So these are some of those genes. If there's a pink box, it means that FOXO was bound very close to that gene. And if it's filled in either green or red, it means that that gene changed expression in response to knocking FOXO out of the genome. And for present purposes, what's important in this is it's called Son of Sevenless. I can explain afterwards if anyone's interested why it got such a ridiculous name, but it's a key component of the RAS signaling pathway. And it turns out that FOXO both binds to it and suppresses its expression. And this really interested us because it gave us a hint that the RAS pathway might be important. So we set about testing it directly. Again, the work of two postdocs, again, Nazif, who I've just shown you, also Kathy Slack, who's just started up her own lab at Aston University. And what they asked directly was, okay, if we look at this protein Chico, its structure is shown here. It can signal down these two different branches. What happens if we knock out its contact either with this branch or with this branch, compared with knocking out Chico completely? Does it matter whether Chico can communicate with PI3 kinase? Does it matter whether Chico can communicate with the RAS branch for the extension of lifespan? So what they did was to start with flies with no Chico, and they put back either Chico itself or a version of Chico that couldn't communicate with RAS, or a version of Chico that couldn't communicate with the PI3 kinase, and looked at what happened to lifespan. Turns out that both branches are extremely important. So this is the control lifespan here in black. If we put back a wild-type Chico, we get a wild-type lifespan, so that shows that the whole thing's working properly. We can then look at the various mutants. The two pathway mutants are here, and Chico itself is shown in red. These are not actually significantly different. So basically, if Chico loses its contact with either of those branches, you get a long-lived fly. In one case, FOXO becomes activated. With the other branch, a different transcription factor gets activated, and it turns out that they talk very intimately to each other on the genome in ways that we're trying to work out at the moment. But what that means is we've got drugs. And I should have pointed out, and I'm sorry I didn't, that we've got this drug, trametinib, which acts against this crucial part of the pathway. And trametinib is used in humans for treatment of melanoma. It's an anti-cancer drug, it's a GSK drug, and it's a very potent inhibitor of this ERK kinase. Of course, it was developed against the human ERK, and the fly ERK isn't identical, so we needed to work out whether it would actually inhibit the fly ERK. So this is where it acts in the pathway. And what we did was simply to get some fly cells and give them trametinib and look at how they responded in terms of their signaling. And as far as ERK goes, the crucial information is in this line here. So if we insulin stimulate the cells, when there isn't any drug present, what we see is this nice fat band of ERK it's a phosphorylation of ERK. And if we have a very low dose of trametinib over here, we see some phosphorylation. But if we up the concentrations of trametinib, we lose it completely. So it is an ERK inhibitor in the fly. And we also need to be sure that it's only inhibiting nothing else crucial that it might be in the pathway. And these are two candidates here, AKT, which is in the canonical branch, and S6K, which is an output of the TOR kinase bit. And in both cases, their phosphorylation is completely unaffected by the drug. So it's potent and specific. And if you give it to flies, it increases their lifespan if you do it at the right dose. So what we have here are 3 different doses: purple low, red medium, blue very high. And you can see that the low and the medium increase lifespan in a dose-dependent way. The high dose is interesting. It kills young flies, but if they make it through to about 60 days of age, then there's actually an increase in their maximum lifespan. And we're doing some experiments now where we start feeding it to the flies only when they're old. And it looks as though a high dose, but at old ages, is the best way to do it. But we now have, again, a nice chemical probe for this branch of the pathway, and because these 2 transcription factors cooperate with each other so heavily, we've probably captured most of the insulin signaling effect. And finally, we have this drug lithium, which is just coming to light at the moment. So lithium is used for treatment of bipolar disorder in humans, and what it does is to inhibit this gene, which is called shaggy in the fly. It's the GSK3, for those of you who know about this, which talks to yet another transcription factor, this cap and collar transcription factor, which is known to be very important in various forms of cell stress resistance against electrophiles, oxidative stress, and so on. The cap and collar becomes activated under those stress conditions, and in the fly, lithium clearly activates it by inhibiting shaggy. The reason we got interested in lithium was there was some very nice data from the worm showing that it could produce a dose-dependent increase in lifespan, shown here, maximum lifespan at 10 millimolar. Also, there's rather nice demographic data from humans, although of course it's purely correlative. So these are Japanese populations living in different prefectures, and what's plotted here is simply all-cause mortality against the concentration of lithium in the drinking water in that prefecture. And there's this really remarkably strong negative correlation with quite a large sample size. So mortality is lower where the concentration of lithium in the drinking water is higher. And these aren't suicides, this is all-cause mortality, so it's basically various forms of age-related illness. So we decided to look in Drosophila and see if the effect is evolutionarily conserved. This was done by a PhD student in the lab. Jorge was now actually a postdoc in Boston. And he found that the situation is very similar to that in the worm. There's a dose-dependent increase in lifespan as long as you don't overdo it, up to 5 or 10 millimolar, but then the drug becomes very toxic indeed. And this is very well known in humans as well. There are nasty side effects of high lithium concentration, particularly in the kidney. And so as long as you keep within the dose range here, flies and worms look very similar to each other. So now we've got the 3 drugs for the different bits of the network, and we can ask the important question, does it matter which bit you inhibit? Are these things acting to some extent independently of each other, or are they all actually converging on the same thing downstream? So inhibiting just one is as good as inhibiting all 3. So Jorge's been looking at this. We're talking about unpublished data now. So one of the first combinations he looked at was lithium and rapamycin together. And what he found was they were acting quite independently. So they basically add together to increase lifespan. If you give the flies the dose of rapamycin that produces the maximum increase and then add on the lithium, lifespan goes on up. And it goes on up by as much as if you give them the most effective dose of lithium alone. So at least at the lifespan level, they're acting quite independently. But interestingly, they're not when it comes to what one might call side effects. So rapamycin, at the maximum effective dose, actually makes the flies fat. They're carrying more lipid in their fat body. But if you combine that with lithium, although it still causes the increase in lifespan, it brings the lipid phenotype back to wild-type levels. So they neutralize out each other's side effects, which I think is quite interesting. And if we do all the possible combinations, so the 3 single drugs, the 3 double combinations, and the triple combination, then you get a real forest of survival curves shown here, with this red thing, the winner, and by fly standards, this is an absolutely enormous increase in lifespan. And what we have here is the triple combination of low-dose lithium, most effective dose of rapamycin, and most effective dose of trametinib. So what these data are telling us, I think, is that if this translated to mice and to humans, we would be looking at a polypill. There's more than one underlying aging process here, and we can inhibit them independently, and we can actually show at the signaling level that the effects of these drugs are largely independent. They don't cross-react with each other's targets. So almost certainly for humans, we would be looking at more than one drug in any preventative kind of pill. And actually we have some indications now that trametinib extends lifespan in mice, as does rapamycin, and we're just starting to look with lithium as well. So it looks as though these effects are evolutionarily conserved. So we'd like to really nail that one and be sure about trametinib and lithium in the mouse. It would be very nice to know how many underlying axes of aging we're talking about here, and each of them can have quite broad spectrum effects on health during aging. And the ultimate aim here, I think, is to design a polypill to ameliorate aging-related decline in humans. So I think the way that a lot of people in this field are starting to look at things now is this. If we think about aging-related diseases, that some of the classics are here, there's been a very successful agenda of studying them pretty much independently of each other and in different research institutes, people going to different conferences and so on, and the clinicians who deal with the different diseases being largely independent of each other as well. These are different medical specialities. But I think there's a big push from the aging community now to try and add on a new way of thinking about things that may be helpful, which is that we may be able to intervene in these underlying aging processes themselves and produce this broad spectrum improvement. And we're also talking about prevention rather than dealing with the disease once it arises. I mean, some of these interventions are effective in disease models, but they're much more effective at stopping the disease from starting in the first place. And with that, I'd like to very gratefully acknowledge the people who I've worked on with this all over the years, and also our very kind funders. And I'd like to thank you for your attention after lunch, and I'll happily take questions.
Thank you very much, Professor Partridge. We are going to have a panel discussion, but I would just like to ask if you have some specific questions to Professor Linda Partridge. Yes, over here.
Yes?
How often do you give these drugs to the fly within which span?
So we always start in adulthood. We let them come through development normally. And then in both the flies and the mice, when we're doing drug studies, we let them mature. There's always a maturation period after full growth when the reproductive organs are growing and so on. And then we start after that. So it's usually at about 4 or 5 days in the fly and about 3 months in the mouse. But it's more complicated than that because there are real timing effects with these drugs. So for instance, we've discovered recently with the fly that rapamycin is acting in a very narrow time window at the beginning of adulthood to exert its effect. You can put them on rapamycin for 2 weeks, take them off, and get the full effect. So we're trying to figure out how that's happening at the moment. And in mice, because rapamycin can have side effects, so that TOR that I mentioned, it's actually present in 2 protein complexes, and rapamycin inhibits both of them. And one of them seems to mediate the beneficial effects, and the other seems to mediate the undesirable side effects, which are basically immune suppression and insulin resistance. And it turns out that if you feed the rapamycin to the mice intermittently, you can inhibit the good complex, but not the bad complex. So timing does matter. But with the flies, when we give them the drug, we usually give it continuously. It's just in the food. It's actually, with at least 2 of the drugs, probably the most vulnerable organisms can be the kidney rather than the liver. But what we're okay, so going through each one. Lithium is being given at a dose. If you look at the serum concentrations in the mice, they're well below the human treatment levels. And also, if you look very closely at the histology of the mouse kidney, there's no effect at the doses that we're using, so it's low dose. Trametinib, we're at something like 100 times lower than is used to treat melanoma. And rapamycin is more like treatment levels, but as I've said, intermittent is the way to go with that. So these are all lower doses, basically, than you would use to treat a disease.
Any more questions?
If you think these drugs will become used on humans, when you think they will be available and common in public? What is the time
I think
span?
after I think there needs to be a lot more certainty for each of them. So with lithium, we don't yet know whether it extends mouse lifespan.
Okay.
So we don't know whether that effect that's so clear in the invertebrates is going to be there in the mouse. I mean, we're talking about targets that are involved in cellular stress resistance, which is almost certainly a very major feature of life for those two little invertebrates. Maybe less important in mammals in a day-to-day sense. So I think the jury's completely out on lithium. On the other hand, its safety profile is very well known in humans because it's been used for so long for bipolar disorder. Trametinib is an almost completely new drug. It's only been licensed, I think, for 2 or 3 years. So there's a lot more work to do with that, particularly in mice as well. Rapamycin, I think, is pretty well understood, and small-scale clinical trials are starting in humans. So Novartis did a trial recently. So I should back off. There was a preclinical trial in mice showing that old mice are like old people. They respond very poorly to vaccination against influenza. But if you give the mice a pre-treatment with rapamycin, then you can bring their response up to the level of youthful mice. And it turns out to be to do with, it's the hematopoietic stem cells and their response to the inoculation. They become more responsive if you pre-treat with rapamycin. Novartis essentially did exactly the same experiment in humans. They didn't follow up looking at response to experimental infection, obviously, but what they did do was to look at the immune profile of the people who either had or had not been pre-treated with rapamycin, and it clearly potentiated the immune response. That's published. So there are other similar studies going on. There was another looking in California at a group of older men, and they looked at effects on mobility and progression from mild cognitive impairment, and again the trial was licensed. There's also a drug called metformin, whose targets are much less clear than the targets of the 3 that I'm talking about, but the FDA have actually agreed to a very interesting clinical trial with metformin. So it's the first line of defense against type 2 diabetes these days. It's used universally, so its safety profile is very well known, and it's an extremely safe drug. And what the FDA have done is to license a trial not for aging itself, because aging is not recognized as a disease by the FDA yet, although I think that may change, but against a very wide spectrum of diseases, so including cancer, cardiovascular disease, obviously diabetes, and 2 kinds of neurodegeneration. So slowly it's coming.
Thank you.
Yeah. From what I saw, it was something like a 50% increase in life expectancy with the flies with all 3 drugs. Is that
I think
correct?
it's more like 40, but it's big,
40.
yes.
It's obviously quite hard to measure quality of life in flies, but if you had to make some kind of an assumption, what do you think that this increased lifespan does to flies, and what would that mean if we continue to clinical studies?
Well, again, this is something that we've really got to do the work on. So we're pretty confident that rapamycin's doing something very similar to dietary restriction in the gut. But the fact that the other 2 drugs are acting independently in terms of targets and lifespan extension argues that they're going by different mechanisms. We don't yet know whether trametinib has sexually dimorphic effects. The data I showed you were for females. We don't know yet whether it extends male lifespan or not. And lithium clearly extends lifespan of both sexes equally. All 3 drugs go across the blood-brain barrier, so there may well be things going on in neurons, and we need to look at that very carefully. We need to do a lot of behavioural testing, looking at learning and memory, the senses, and so on. And the work just hasn't been done yet. So I think they're going to be going for very different things, but apart from this gut rescue, I don't know what the others are going to be.
Something left for our generation then maybe. We'll see.
Yeah.
Thank you.
Come in.
Yes. Well, we heard a lot about FOXO today. We in a lot of these different talks, and I'm wondering, would it be able to synthesize FOXO specifically, and if a human takes it specifically, would that increase a human's lifespan?
So your question is if a human did what?
FOXO. If you increase the concentration of FOXO in the human body, would that increase the lifespan of a human?
I can't answer that question, and nobody's done it in mice. I can tell you that if you overexpress FOXO in flies, as long as you don't do it in the wrong tissue, so if you do it in the brain, terrible idea. But if you do it in the gut and the fat body, which is the equivalent of the liver and white adipose tissue, you do get a long-lived fly. Yes. But what FOXO is doing when it gets into the nucleus, so the way that FOXO is regulated by the pathway is through nuclear localization. If you knock down the upstream activity, FOXO goes into the nucleus. But it's not clear that all of what it does there is to do with directly itself acting as a transcription factor, because you can make DNA binding mutant. So the way that the structure by which FOXO binds to DNA is very well known. And you can therefore work out what the critical residues are, and you can mutate them, and therefore have a transcription factor that's there in the nucleus, but it can't bind to DNA. And you still get some of the effects of FOXO with that mutant thing. So it's almost certainly interacting probably with other transcription factors, perhaps even the one from the RAS pathway, and acting as a cofactor of some of its critical effects. So the situation in humans is going to be much more complicated because there are 4 FOXOs, which are to some extent tissue-specific, but if you start messing around with one of them, the others may compensate. I think we've got to sort this out in mice before thinking about people, really.
All right, thank you.
Linda, beautiful presentation. I was wondering, actually the fly would be the best place to start treating older flies. So there's of course a lot of discussion ongoing that all the medication is always tested on younger individuals and is never completely fit in humans for a 70-year-old. So what would happen? I mean, these drugs you could test on a fly that has never seen the drug, but then if you would give it in the last third of life, I think it would be a great indication.
I think it's
of
very
the
interesting.
timing.
You can do that with mice as well. So, and I think probably just as effectively, I think they're both very good for that kind of question. So when we discovered the trametinib effect in flies, we actually had a cohort of really old, elderly mice, and we managed to get permission in time to feed them the drug, and it actually showed a dramatic increase in survival in the males, not the females. That may be a dose effect, I don't know. We could only go with one dose. We didn't have that many mice. But yes, you're right. And I'm pretty sure with dietary restriction, Walter would know a lot more about this, that actually the way to implement is probably age-specific. In flies, if you really want to maximize their lifespan from dietary restriction, you have to ease off as they get older. Nobody's really looked at that in mice, but I bet it's the same. If you've got an old, frail mouse, probably needs a bit more.
Okay.
No, so I think they're very interesting, these age-specific effects.
Yeah, yeah.
And also, in a lot of clinical trials, females are underrepresented as well.
Yeah,
For
exactly.
all kinds of...
Yeah.
So we have very little information about older females.
Yeah, so if you can show to the pharmaceutical industry how big the effect is of age and sex in the animal studies, I mean, that would really improve the situation for humans for more testing.
Yeah.
Yeah.
Thank you very much, Professor Partridge.
Thank you.
Please take the seat in the panel.
All right.